communities and the habitat that we all know and love here on our part of the coast. So I'd just like to present our speaker tonight, Zachary, who is a fifth-year PhD student in the Department of Integrated Biology at Oregon State University, where he is also co-advised by Bruce Megan and Sarah Hacker with the fellowship support of OSU and the National Science Foundation. Zach studies rocky intertidal ecosystems of Oregon, California, and Nova Scotia. His interest in how climate change, disturbance affects disease and other species. Continue on, I know I'm pretty loud anyway, so. Cool, all right. All right, so anyway, as I was saying, his interest in how climate change disturbance affects disease and other species interactions influence the dynamics of community succession. Following his passion for conservation, Zach also published research on marine protected area comments for commitments and unintended consequences of biocontrol insects for threatening wildflowers. Prior to his doctoral studies, Zach completed his BA in biology and environmental studies at Lawrence University, where he was elected to Pi Beta Kappa and graduated summa cum laude in 2015. While at Lawrence, Zach was awarded various scholarships as well as an opportunity to study abroad in Madagascar. And in recognition of his commitment to environmental issues, he brings a wealth of conservation understanding as well as an enjoyment of birding, long distance running, and backpacking and playing with his cat Kamala. So, with no more, I'll let Zach take it away.
Can you all hear me okay? All right. Thank you, Kayla and Graham, for the introduction, and thank you, everybody, for coming here tonight. It's a rare opportunity as a PhD student to be able to talk about your research in front of folks in a non-academic setting, and I really appreciate this opportunity, and thank you to the sponsoring organizations. So as Graham talked about, I study rocky intertidal community ecology, and I'll give you a brief overview as to what that is. This is a photo of me doing field work at one of our field sites in southern Oregon, near the city of Port Orford. And in the background, you see these four large rocks. That is the Redfish Rocks Marine Reserve. So I work in close association with marine reserves, although I don't actually work in the marine reserve because they're all offshore. So this is a beautiful field site, and I'm very lucky that I have the opportunity to study such amazing places like these. Before I get into the nitty-gritty of my talk, I just want to give a brief land acknowledgement that Astoria is located within the traditional homelands of the Kathlamet, Clatsop, and Chinook peoples. And today, living descendants of these people are part of the Confederated Tribes of Grand Ronde, Community of Oregon, as well as the Confederated Tribes of the Siletz Indians. All right, so the rocky coast of Oregon is a place that we all hold near and dear. It is a place that I return to at several points during my life. When I was a little kid, I had the opportunity to go to Cannon Beach, and I'm actually from Wisconsin. So traveling all the way from Wisconsin to Cannon Beach, Oregon was quite a road trip for me and my folks and my brother, and I fell in love with the tide pools when I was a 10-year-old. And again, when I started college, I actually spent the summer, for a week of the summer before I started college here on the Oregon coast. And I really enjoyed that. That was when I was first starting to do bird watching and first learning about ecology, and I really fell in love with the tide pools. And I never thought at that time that I would come back here and make this wonderful place my home, but I'm very happy to be here now. And I think the Oregon coast is a very special place. It provides us with inspiration, just the opportunity to go out and experience the ocean. It's such an enormous, beautiful part of this planet, and having that on this coast is a real blessing. I moved all the way from Wisconsin because of the ocean here, and it's something that I hold near and dear to my heart. It's also a place of recreation. We find ourselves walking along the beach, following the swash line, thinking about what organisms live in these habitats that are so rugged. It's also a place that provides a lot of tourism to the state of Oregon. Many people come from California. Actually, my high school biology teacher has contacted me a few times over Facebook saying that she wants to come to Oregon. She wants me to host her. She wants to come all the way over from Wisconsin to be a tourist here, and it's such a special part, and we have it here in our backyard. The Oregon coast, and especially the rocky reefs off coast, provide a lot of habitat for important fisheries, so very economically important species. And finally, the Oregon coast is a place of discovery, and I specifically work with rocky intertidal communities to do scientific discovery. And so today I'd like to talk a little bit about my research. And first, I'd like to give you an introduction to rocky intertidal communities, if you're not familiar with them. And then we'll go into a description of some changing conditions that these communities are experiencing. And finally, I'll discuss a few of the experiments that I and my colleagues at Oregon State University are doing in the intertidal. So first, rocky intertidal communities are very dynamic systems. They have a lot of different species, and they're organized based on the high tide and low tide marks. So generally, we organize rocky intertidal communities into 3 zones. There's the upper shore, the middle shore, and the lower shore, or as we call them, the high zone, the mid zone, and the low zone. And these 3 zones are really distinct. If you go out at low tide and you look at the rocks, you can see 3 distinct layers of species. And generally, the high zone is only exposed to water during highest tides, only underwater during highest tides. And the lowest zone is only out of water during the lowest tides. And so as the tide goes in and out, there's a great variability that these organisms are experiencing. So generally, the high intertidal zone experiences the most kinds of physical stress, so things like high temperature variation, higher salinity variation, more time exposed to air, and also more sunlight. In contrast, the low zone generally has the greatest species diversity, and because of the number of species that are present, there's also higher competition, and it's where a lot of the major predators live. So predators like sea stars and crabs are also more abundant in the lower intertidal zone. And today, most of the research that I'll talk about is in the low intertidal zone, because that's my particular focus and my favorite zone because of the high species diversity. But I also get really wet sometimes when the waves come in and they crash over me and my plots, so it's kind of risky at times, but we have fun. And I'd like to talk a little bit more specifically about a couple species that are really important in the rocky intertidal. So quick question, does anybody have any guesses as to what a keystone species in the intertidal would be? Okay, so first I'll define keystone species. Keystone species is a consumer that has a disproportionately large effect on communities and ecosystems relative to their abundance. Any guesses? Sea stars, very good. Yes, specifically the ochre sea star. So the Pisaster ocreus sea star is one of the most important, iconic predators in this ecosystem. So my advisor's advisor, so my academic grandfather, as it is, was the person who discovered that keystone species are present in rocky intertidal systems. And he actually came up with the concept of keystone species as it relates to this organism, this sea star, Pisaster. There are also species that are called foundation species, and these are organisms that interact strongly with other organisms due to their high abundance or biomass, and they often create habitat through their tissues. And 2 of my favorite foundation species are surf grasses and mussels. And I won't get into this to the talk today, but I actually have a whole experiment where I'm studying the interactions between these 2 species as part of my dissertation. And then these kinds of species, keystone species and foundation species, are also present in other in other systems, and in the subtidal, so just a little bit farther offshore, the sea otters have historically been keystone species in Oregon, and there's still keystone species in other parts of the Pacific coast. And the kelps are the foundation species underwater. So here we have a photo of bull kelp and also giant kelp, and these really provide a lot of habitat for fisheries and invertebrates and lots of other organisms. So just keep these kinds of species in mind: foundation species, kelps and surf grasses and mussels, and keystone species like sea stars. And this is a kind of a hairy diagram, but fairly complicated. All I'm going to say is that the oceanographic context also matters in addition to the species. And the main thing here is that upwelling is very important, and upwelling strength really affects the rocky intertidal communities. And this is some research that my advisors conducted, and also an experiment that I continued on in my PhD. So why do we study the rocky intertidal? As I mentioned, it's a very dynamic, diverse system, but it's also really tractable from a research standpoint. So if I wanted to study things about ecology in a mountain, I would have to travel many miles from the base of the mountain to the peak of the mountain to experience different types of communities, varying from deciduous trees to coniferous trees, all the way up to lichens and mosses near the peak. But in the rocky intertidal, I can experience this same kind of zonation over a matter of meters. So we can do experiments really easily that allow us to determine how communities are structured and how ecosystems function. So the spatial scales are very favorable in this kind of study system. And the rocky intertidal has lots of different species that are small to us humans, but serve very important roles in the ecosystem. Similar roles to, say, the gumbo chitin is similar to an elephant. So this is a large chitin, the largest chitin in the world, actually. And it is a very large grazer, plowing through, eating algae. So it's kind of like an elephant. Then you have smaller grazers like limpets that are kind of like deer. And also different types of predators like whelks, which are these predaceous snails that drill into the shells of other organisms, and then they slurp out the goo from inside. And then of course there's the Pisiaster sea star, which is the keystone species. It's kind of like the wolf of the intertidal. So we have all these different organisms in the study system. The ecosystem is very easy to study, and these make it favorable for ecological study. And some days we go out and we do research, and it's a beautiful day. Like, this is one of my study sites at Cape Perpetua. The ocean is very calm, nice and sunny. This is at Rocky Point in Southern Oregon. My undergraduate Emily and I collecting data in the field, and my brother was with me and he took this photograph. We're having a grand old time doing research. Some days, however, it's really rough, and it's dark and rainy, and there were 78 earthquakes off the coast of Oregon that day, and the waves are rolling in, and my girlfriend's upset with me because I'm still going out to do field work even though the conditions look like this. And I actually didn't get any work done, but as you can see in the foreground of this photo, I've got a cage, which is one of my treatments where I exclude sea stars and other predators. And I was gonna go down there to take data, but the waves were just too rough. So instead I just took some photos, but I still had a fun time out there in the intertidal. So, given all of this information and how easy the rocky intertidal is to study, my advisor, Bruce, has been at Oregon State for 45 years, and maybe one day I'll be lucky enough to have a career that long. But he's been working at some sites since the early 90s. So this is one of our longest running field sites up in just north of Depot Bay, a place called Fogarty Creek. This photo was taken in 1991. Here's a photo from 2007 with some of the researchers from OSU out in the intertidal. 2009, we're still working there. 2012, 2013, 2017, out collecting data. And generally, this ecosystem seems constant. If you look at these photographs back to back to back, it looks like this system is fairly stable. However, I'm here today to talk about drivers of change in rocky intertidal communities, and I am really a believer in that there is change going on, and so that's what I'd like to pivot to now, to talk about some of the changes that are happening. First, how are they changing? How are these communities changing? And second, what are the ecological and physical drivers of those changes? First, as you are probably well aware, there was sea star wasting disease, which was an epidemic that occurred a number of years ago, but it's still ongoing. This is an article from Hakai magazine, which is an excellent publication that I'd encourage you all to check out. And in this article, they talk about how sea star wasting disease seems to be exacerbated by warm water temperatures. So when the ocean is warmer, we tend to get more of these disease outbreaks. And sea star wasting disease is a progression of symptoms that starts with lesions along the sea star. So this is a photograph taken first day. The sea star has some lesions where the arms meet the body. And then that leads to rapid tissue decay. By day 2, the arms are starting to disintegrate from the body, and by day 3, the sea star is really just a puddle of goo. And this is a very devastating epidemic that affects more than 20 species of sea stars, ranging from the intertidal to the subtidal sea star. And this particular mass mortality event began in 2013 and 2014 on our coast, but it does continue today. Once in a while, I'll go out to the intertidal and I'll see a sea star that has wasting and I'll document it. Another stressor that these communities are experiencing is extreme heat stress. So last summer, you all remember the heat dome, which we experienced, especially where I live in the Willamette Valley, but a little bit less extent here on the coast. And this photograph was taken up in British Columbia, where this severe heat dome caused the death of billions of organisms. In this article from The Atlantic, they interviewed a professor at the University of British Columbia, and he was talking about how the low tides were during the afternoon, during the highest temperatures of this heat dome, and it just cooked all of the mussels and sea stars and the associated organisms. And this is a really tragic event. Luckily, most of the Oregon coast escaped the effects of this heat dome because our tides are a little bit earlier in the day. The low tides are earlier in the day. But there are also heat waves that occur in the ocean, and these are known as marine heat waves. A marine heat wave is defined as a prolonged, extreme oceanic warm water event, and they can persist anywhere from weeks to years. In 2014 to 2016, we had a marine heat wave event known as the blob, which really was a warm water mass that sat off the coast of Oregon and California for a number of months. And it caused mass mortalities of seabirds like common murres and kelp offshore. And it also killed intertidal kelp, and that's part of my research, as I have a long time series. And I'm looking back in time to see to what extent the marine heat wave affected these communities. Unfortunately, marine heat waves are growing phenomena. They're becoming more common, and they're really globally spread. So this is, I'm not going to go into each one of these, but there's many different heat waves events that have occurred around the globe. The blob was just one of those. And these heat waves are becoming more common through time. So this is a graph showing a number of different climatological models, and over time from 1900 to 2020, there are more marine heat wave days on average per year. And the projection is that these marine heat waves are becoming the new norm. Into the future, the majority of days may be marine heat wave days, as is relative to 1961 to 1990. And then in addition to heat waves, we have other kinds of stressors. So I'm going to show you a series of photographs from one of our field sites in Port Orford Heads State Park. This photograph was taken in June of 2020, and as you can see in the foreground, there's a really dense, thick mussel bed. So all of the black is Middleus californianus, which is a type of mussel, the large California mussel. And this is the major dominant organism in the intertidal. And it's also the preferred prey for the predator Pisaster opretius, the sea star. In July of 2020, you can see that there's more sand building up around the edges of this mussel bed, but it's still largely intact. But by October of the same year, nearly all of the mussels had disappeared. And I didn't know, taking these photographs, that I was going to document a change like this. I just go out and I take photos before I do my field work, just to say I'm at this site, so every photo is from this site. And that absence of mussels persisted into May of last year. And I haven't been back to the field site yet, but I'm expecting there to be a lot of bare rock still, which is crazy because the mussel beds used to be like at least a foot thick, and now they're just all gone, all wiped off the rocks. And some of my plots are actually under sand, so this is a photograph from the same field site looking down coast. And this sandy patch didn't used to be there. I used to have to jump from rock to rock in order to not fall in these channels that are there in order to survey my plots. So this sand burial is another stressor that these organisms are experiencing. And given all these pressures, so we've got sea star wasting disease, the marine heat waves, and sand, some new research from my lab has come out, and this was just published in January of this year, showing that the Oregon coast, the rock intertidal in particular, is becoming what we call subtly destabilized by the pressures of climate change. And I'll go into what I mean by that, so that subtly destabilization is a little bit of a hairy concept that I'd like to break down with some ecological theory. So a little bit of a scary graph, but we'll go through it slowly. So in ecology, we like to use models in order to try to break down a more complicated concept. And one of the models that we like to use is called the ball and cup model. So you can think of the ball in this diagram as different community states. So in one state, we might have a coral reef. In another state, we might have a macroalgal reef. In one state, we might have a kelp forest. In another state, we have an urchin barren where the urchins have chowed down all the kelp and there's nothing left. So these are different types of community states. And they exist under different environmental conditions. So in this model, the cup is the environmental conditions. And communities can shift from one stable state to another stable state as a result of different changing conditions or different changes in species. And if they do this major shift, which is called a regime shift, they might cross what we call a tipping point or a critical threshold between different types of states. And as I said, there's two different ways that this can happen. There could be a shift in the variables, which is a shift in changing species, or the parameters, which are the changing environmental conditions, can happen. And that's more of what climate change is doing, is changing these conditions around us and then causing communities to shift in response to that. And if a community is existing in one state, we might want to predict what it might look like in the future. So this is a figure from my advisor's paper that just came out in the Proceedings of the National Academy of Sciences. And here we have these idealized communities over time, and they experience different disturbance events, so 1, 2, and 3 in this diagram. And every time a disturbance event happens, it removes species, it kills something. And sometimes the system is able to bounce back from that disturbance, and we call that kind of system resistant. Other times the system takes a while to recover, but if it recovers eventually, we call that system resilient. But if the recovery slows over time, that's an example of diminishing resilience. So the community is becoming less and less resilient to these kinds of perturbations over time. And there are two different cues that I want to highlight as to what might be an indicator of diminishing resilience. The first is that we see a decline in the mean community state, and we could also see an increase in the variability of the community state. So both of those things might indicate diminishing resilience. So how do we test this in the field? Well, we design an experiment, and sometimes we design experiments, but we don't really know what will happen, but we just keep doing them because we like doing them, as is the case in a lab that's been around for 45 years. So this is an experiment that was started many years ago, but I'm going to show a few photos of our plots. And this is from 2012, so we have 2 different experimental treatments. In the first treatment, we have what we call a control plot, which we never cleared. And then in the second treatment, we have a disturbance plot, which we clear annually. And in both of these plots, we track the community composition, which means what species are there, at what site, and at what time. So we take these photographs frequently, monthly if we can, especially during the summer. And then at the end of the year, we take a final photograph and then we clear the disturbance plot, but we leave the other plot just as it is. So as you can see in the top row, we've got the control plot, it's surf grass, each of these photos. In the last photo, it's still surf grass, and that's what we want to see. This is an intact community. It's very resilient. Whatever kind of disturbance events don't really affect it. But this disturbance plot is something that we are actively changing, and then we allow species to come back over time. And we find, when we're looking at these 2 different treatments, different patterns. So in the top row here, we have the community structure, the mean community structure. And on the left-hand side, we have the disturbance treatment, and on the right-hand side, the control treatment. And we see a negative slope in the disturbed plots. And as I said, the declining average community structure is indicative of diminishing resilience in this community. And then if we look at the variability, we see an increase in variability in both the disturbed plots and the intact plots. And it's interesting that we see this in the intact plots because they're supposed to be the control. They're not supposed to change that much. But we still see an increase in variability there. And as I said, the increasing variability is another indicator of diminishing resilience in this community. So what caused these changes? We can do some statistical modeling and other types of analyses in order to determine the causes or the likely causes. The indication is that it's likely large-scale changes in physical and ecological conditions which occurred during the course of this experiment. So this was from 2012 to 2019, and that included both the marine heat wave and the sea star wasting disease epidemic. As you can see from this image, there was a lot of warm water off the coast of Oregon and California throughout this period. This is a graph that just shows sea surface temperature anomaly. So warm colors mean warmer than average. And temperature, especially hotter temperatures, were the strongest driver of these changes, and particularly the marine heat wave event, but also there was an El Niño event, and El Niño is when there's warmer water than normal, and that occurred on top of the marine heat wave in 2015 and 2016. There was also intensifying upwelling, which I'm not going to go into too much detail, but I'm happy to answer questions about. And then, of course, the predator loss due to sea star wasting disease. So we might ask, given these different conditions, we saw important physical conditions and important ecological conditions, what is their relative importance? Which one is more important than the other? And how might we adjust management to compensate for that? And then also, how does this vary along the coast? Because the different parts of the Oregon coast experience different oceanographic conditions. So my advisors designed another experiment, and they actually started this experiment in 2006, which was when I was in middle school, but I'm lucky enough to take it over as part of my dissertation work because it's still happening now. So it's a plug for long-term data sets. I think they're really important, and you may not know when you start a project how fruitful it will be in the end, but there can be a lot of great findings from them. So anyway, this experiment featured 5 different treatments, 2 treatments that were very similar to the last experiment. We've got a control plot, which we never cleared, a recovery plot, which we cleared initially and then never touched again, and then 3 other treatments where we removed, as often as possible, invertebrates, macrophytes, and consumers, which means herbivores like the chitons, like the limpets, and then also the predators like sea stars and whelks. So we did this experiment at 13 field sites on the Oregon coast and California coast. And I've spent much of my PhD time traipsing up and down, monitoring all these, which has been a super fun part of my job, and I love doing field work. And generally what I do when I go out into the intertidal is I look at these communities and I do what we call a community survey. So I look at the plot and I try to analyze how much of each type of species is present. So in this plot, you can see the grass is the dominant species, and I kind of eyeball it, move things around in my head, and then I think, okay, well that's about 60% cover if I move this here, and then what's the rest of it? There's some coralline algae, some kelps on the side, some foliose algae, and we record all these data. We do this many, many, many times, and I have over 4,000 surveys of us doing this, so lots of data. And all that data shows us some changes over time. And I'm not going to go into some graphs, but I'd like to show you some photographs of our communities. So first, this is from our field sites in Northern Oregon, and this is one of the control plots. So this is a plot that we never changed. We didn't manipulate in any way. In 2006, it was entirely surfgrass. And surfgrass is a long-lived species that is very robust. It doesn't die annually, it's a perennial, and it can withstand a lot of different disturbances. By 2014, 8 years later, it's still there, but you can see in the upper right of this photograph, there's some mussels that are starting to encroach. By 2018, the mussels have entirely taken over this plot. And between these two photographs, in 2014 and 2018, you can see the gradual progression of mussels taking over. And we believe this is due to the sea stars disappearing, at least large sea stars, from this habitat. Fortunately, they have now rebounded, but my hypothesis is that the mussels are still there in 2021 because they're now so big that the sea stars can't eat them anymore. And one thing I'd like to do this summer is actually measure how big they are and see if I can test that hypothesis. If we go a little bit farther south on the Oregon coast to Cape Perpetua, this area is traditionally dominated by more invertebrates. So you go out and you see lots of barnacles and mussels. And in 2009, this is one of our recovery plots. So we cleared it to bare rock in 2006. By 2009, 3 years later, it's covered with barnacles. 2014, still covered with barnacles. And then sea star wasting disease hit. And sea star's second favorite food item are gooseneck barnacles. In 2018, they're just covered the plot. It was so bad that I had to go out there 2 days in a row to try to find this plot. And I'm traipsing around there with a metal detector trying to find my bolts in the intertidal. And it was just so covered with these gooseneck barnacles. But the sea stars are starting to rebound, and they're eating the goosenecks at this site. Go a little bit farther south along the Oregon coast to Cape Blanco area. This site is a mix of invertebrates and algae. In 2009, this plot is still mostly bare rock. 2014, though, it's covered by different kinds of algae, different kinds of red algae and brown algae. And then sea star wasting disease happens, and suddenly we see all these mussels there, where there were never mussels before. And again, this is the favorite food of sea stars. But with sea stars starting to rebound, at least here, now we have surfgrass taking over. So this is a very variable plot. This individual plot is highly dynamic. And finally, this is my southernmost field site in California, just north of Fort Bragg. And here we have a plot that's dominated by kelp, specifically sugar kelp. And most of the California sites have historically been dominated by kelp. In 2014, kelp was still really plentiful, but kelp is also very susceptible to heat stress. And so with the marine heat wave of 2014 to 2016, combined with sea star wasting disease releasing mussels of their major predator, mussels were able to take over this space once the kelps died. And now they're so dense in 2021 that I can't even find the plot very well. So, given all of these data, I'm able to make a plot of community variability over time. And you'll see a few things. You'll notice a few things about this plot. First, there's a major jump in the first 3 years in variability. And that's likely due to early succession. So as I said, most of these plots were cleared initially, and then species are trying to reestablish themselves. After that, though, there's a pretty flat period for 6 years, followed by a jump and another flat period for 6 years. And this increasing variability is likely an indicator of diminishing resilience in a different experiment from the one that I showed earlier. So all of this together can allow us to project into the future somewhat. And there is evidence of community recovery at some sites. The sea stars are starting to come back. We're getting bigger and bigger sea stars. I have only ever done research after sea star wasting disease, and I'm starting to notice, wow, there's way more sea stars out here than I've ever seen before. And then I asked my advisor, Bruce, who's been here since the 1990s, like, Bruce, how many sea stars is this compared to what it used to be? And he's like, this is nothing. There used to be so many you couldn't even step without stepping on one. And that is crazy to me. So we're approaching what we had before in the early 2000s, but it's nothing like it used to be. So in the short term, this kind of destabilization of local communities is likely to continue. As I showed, marine heat waves are probably going to become more frequent and more intense. And that may trigger additional outbreaks of sea star wasting disease and maybe even other marine diseases that we have not experienced in the intertidal yet. And then in the long term, climate change may irreversibly alter these habitats. Sea level rise, if it happens on the Oregon coast, will inevitably push up the intertidal organisms because the tide line will just be higher, and the organisms will have to move in response to that. And warmer oceans may favor sessile invertebrates over kelps. So as we saw following the marine heat wave, invertebrates were able to take over. So in all of these kinds of changes, there are winners and losers in the communities, and it's part of our role to document these kinds of changes. So with that, I'd like to acknowledge a few people that are really fundamental to me being here. First of all, my advisors, Sally Hacker and Bruce Mengi, who've been at Oregon State University for a long time. And then also my funding sources from the Ecological Society of America, the National Science Foundation, and Oregon State University. And then also all the folks that I've had the privilege to do field work with for the past few years. So there's me and my brother on the lower left, and some of my undergraduates, Lily, my friends, Roy and Laurel, another undergraduate, Emily, and my friends Dave and Chris, who've helped me do field work. I've spent almost an entire year of my life doing field work, and it would not be possible without lots of teams of people. So we do nothing alone. And then I'd like to thank all the organizations and sponsors who are putting on this wonderful series of talks, and I'm really happy to be here. And with that, I'll take your questions. Yeah, so that site in particular, can you all hear me if I don't use the mic? Okay, so that site in particular is adjacent to a sandy beach, and it's a very narrow area of rocky intertidal habitat. So as the waves come in, and as waves get stronger during the winter with all the storms, bigger swell, that tends to push more sand up into the rocky intertidal habitat, and it really swamps them, and that's why I think we saw the die-off as we were coming into fall and then into the winter there. And I've seen some sites, though, that are much wider intertidal habitats that are also covered by sand. So I think it's really just adjacency to a sand deposition and how much the waves move in the right way to deliver that. And then usually with the really strong storms in the dense part of winter, like January, February, then the sand gets blown out again. So by the time we go out there in April, the sand is mostly gone. Otherwise, I wouldn't be able to find my plots because they would still be under sand. Yeah. Did you have a question?
That was actually my exact question.
Okay.
Yeah, I was thinking mostly about in central Oregon coast,
Mm-hmm.
in the cities, there was maybe
And most organisms can't really withstand that much sand deposition. However, anemones can live they're basically immortal. Like, they can survive sand burial, they can survive pretty much anything unless they get ripped off the rocks. Yeah. Does anyone else have a question? Yes.
At the end, when you said back in the 90s or whatever, that you couldn't walk for stepping on sea stars, is that a stable condition? That sounds like an accident waiting to happen, that they're so overcrowded.
Yeah. Yeah, that's a good question. I should make it a little bit more specific. At some sites, it was like that. The sites on Cape Perpetua where there was so much food available for sea stars that their populations were allowed to grow that high. And I saw photos from that period of time, and it was just like masses of sea stars that were all, one giant pit. And I think the populations had lived at that peak food abundance for so long that it had reached a stable state. So at those sites specifically, yes. But at other sites, like in northern Oregon, southern Oregon, even in California, those are generally dominated more by algae. And if they're dominated by algae, then there's not as much food for those sea stars. So it is variable. It depends on what part of the coast you're talking about.
Well, will that mean, could some, what shall I say, some problematic event arise at there where they're carpeting the seafloor, so to speak? That could some disease or something start there and spread?
Yeah. I think it's interesting, the progression of sea star wasting disease started in California and Oregon, California and Washington in 2013, but it didn't jump to Oregon until 2014. But it did spread rather quickly there. And I think that the proximity of sea stars to each other probably affected the disease transmission. But that's really hard to investigate, especially in seawater. And a lot of these communities are underwater for so much of the time that it's hard to run studies. Like, I can only do field work during a 3-hour window, and beyond that, we can't really study that very well, the disease transmission.
Okay.
Yeah. Did I see a question in the back? Yeah.
Yeah. So a number of major contributors to the community composition of these areas is larval recruitment, and a lot of people are involved with it. Have a larval phases. Each month in the ocean will recruit. And I was wondering, you guys are measuring recruitment patterns of different invertebrates, and how does especially the heat waves affect that, and new juveniles that land on spots, they will get off.
Yeah, I'm really glad that you asked that question, actually, because I have a whole dissertation chapter where I do nothing but count barnacles. And I and my undergraduates were counting these barnacles on settlement plates. So when barnacles are larvae, they are out in the open ocean. They have these swimming planktonic stages, and they feed in the open ocean, and then they recruit to the rocky intertidal systems, and they become adults. And once they're adults, they're fixed to one place, and they're there for the rest of their lives. And we put out these plates that are covered with this kind of rough tape, and the barnacles choose to settle onto the plates, and then we count them and how many there are of different species and in what life stage. And we can see different patterns in the invertebrate larval recruitment, as you're asking about, at the different sites. And the sites in Central Oregon, especially Cape Perpetua and Seal Rock, tend to have the most barnacles. I know because I've counted hundreds of plates from those sites. And the other sites in Northern Oregon tend to have fewer barnacles. And then there's also some sites in Southern Oregon that have fewer as well. And actually, my undergraduate researcher, who is one of my mentees at Oregon State University, her project is going to be looking at the marine heat wave effects on larval recruitment. So I'd love to answer that question. I'll be able to in a few more months. She's going to give a poster presentation on that in May, but we don't have the answer to that quite yet. My guess is that larval recruitment declined during that period, at least for barnacles, but I don't know for certain yet. So we'll have to get back to you on that. Any other questions? Yes?
So, slightly different topic, but plastics having an effect on the systems?
That's a good question. I don't know that much about marine microplastics, but I have seen very little plastic debris in the intertidal, and I've done field work for 5 years now. And occasionally we'll find some plastic debris, like one year I found a laundry detergent bottle that looked like it washed up from China because it had Chinese characters on it. And so these plastics are traveling a very long way to get to our coast. But it depends on things like the fragmentation of those plastics. Generally, the really tiny plastics that can get into tissues are the more dangerous to organisms. And the larger plastics, like that laundry detergent bottle that I found, that's really not going to affect that much unless something ingests it. Like if a turtle eats a plastic bag, obviously that's going to kill the turtle or be very harmful to it. But for our organisms, they're not really feeding that much on these larger plastics, but they might be feeding on the smaller plastics, especially mussels and barnacles, which are filter feeders, and they're filtering out sediments. And another research project that is going on at Oregon State University is we collect mussels annually for a mussel growth project, but now we're starting to analyze their tissues for microplastics. And I know that work is ongoing and hasn't been published yet, and I don't know any of the results, but there will be a publication on these microplastics as well. Anyone else? Yes?
This is, I know, outside your presentation a little bit. You did mention kelp being sensitive to heat, and you mentioned the kelp barrens caused by the proliferation of urchins on the West Coast, and there is efforts going on right now, and even concern about losing our kelp and restoration efforts. I'm wondering how hopeful or hopeless you think that is, or are we just kind of looking with protocols and seeing what's coming to us? Are we just seeing the kelp to be norm? Is there, or should we need to be trying to restore it?
Yeah, I think it is a wonderful opportunity to investigate how important urchins and their predators are in these habitats. So in the kelp forest, as I mentioned, the sea star Pycnopodia, maybe I didn't mention this, Pycnopodia, which is the sunflower sea star, was one of the major predators of urchins. But it was also the sea star species that was most drastically hurt by sea star wasting disease. They are functionally extinct in most of their range now. And with the loss of this sea star, the urchins have been able to proliferate. In Oregon, we also have the loss of sea otters, which were their keystone predator. So between those two predators disappearing in Oregon, we had proliferation of urchins, and they're eating many of the kelps that were along the coast. There are still some kelp forests on the Oregon coast. And there are ongoing efforts to actually cull urchins and do experiments to see the effectiveness of that management practice. So I'm involved a little bit with the Oregon Kelp Alliance, or ORCA, and they just started this past summer doing an experiment at Port Orford and Nellie's Cove, and they've divided Nellie's Cove into two halves. On one half, they're culling urchins, and the other half, they're leaving the urchins intact. And they're going to measure over time the responses of those communities. And I'll be volunteering with Oregon Reef Check in order to assess the communities over time. So I'll be going out and doing these subtidal surveys on scuba and counting the number of kelps and fish and invertebrates that we see in these different treatments. So there is hope in that this kind of practice, culling urchins, might be effective to restore kelps. But it's likely that if it is effective, it's going to need to be done regularly until we can reestablish these predators, the sunflower sea star and the sea otters. Because in essence, what we're doing is acting like predators. When we remove urchins, we are eating them effectively, and that allows the kelp to replenish. Any more questions? Yes?
Do we know what is like some of the driving causes of the wasting disease? Like, was it just the higher temperatures? And if so, what was it specifically about higher temperatures that caused like an epidemic?
That's a really good question, and the disease epidemiology is still being worked out even years later. There are some researchers at Cornell University and a few researchers at Oregon State who are looking at this. And generally, we think it's a species of, or a kind of virus, a densovirus, and it appears to be naturally occurring in these sea stars, but something about the warm water event caused an outbreak of this naturally occurring virus. So maybe the conditions were better, just like we have flu season and cold season when the weather conditions are colder and the virus is able to transmit better. Maybe there was something about the warm water that allowed this virus to transmit more easily or be more damaging to the sea stars when the water was warmer. And that kind of experimentation is still happening, but it's very sensitive because you don't want another outbreak, right? But you have to have these flow-through seawater tanks in order to do experiments and keep the sea stars alive because they're pretty finicky in the lab. I've done some experiments with them in lab, and they all stayed alive, but they required this flow-through seawater tank. And in order to do experiments with diseased sea stars, you have to have this flow-through tank, but you don't want to recontaminate things. So it's kind of hard to do from a logistical standpoint to really understand what's happening. But the leading theory is that it was some kind of densovirus that was triggered to be more damaging during the heat wave. Yeah. Anyone else? Yes?
Full disclosure, I know almost nothing about the ocean, but I do know that there's a lot of invasive plants and animals on the surface. And basically every place you're conducting your experiments. Are you seeing a lot of invasive species in the intertidal zones?
Actually, no. Interestingly enough, I have done some field work in New Zealand where invasive species are more common in the intertidal. So there's some invasive algae that are more abundant in the intertidal in New Zealand, but in the Oregon coast, there really are not very many invasive organisms. And I think invasions depend on two things. Mainly, getting to a spot and having that spot be the kind of environment that you can inhabit. And so the first part, the transmission, depends on people like us or the ocean currents visiting the intertidal and bringing an organism to the intertidal. And then there has to be some kind of space available for that in order for it to establish. And if that space isn't present, then the invasive species can't establish and perpetuate. So I haven't experienced it, but it's not outside the realm of possibility. Any other questions? Oh, sorry. Yes.
Have you heard any conversations about how it affects like regular vertebrates that and feed on the tides here? Like gulls.
I like gulls.
Yeah.
Actually, one of our field sites we nicknamed Gull Haven because there are so many gulls there.
They eat a lot, so
Yeah.
there's benefit.
So the vertebrates generally are feeding on the invertebrates. And if sea stars have declined and their invertebrate prey aren't being eaten by the sea stars anymore, then the invertebrates become more abundant. And that would generally benefit the vertebrate predators. So the species like gulls, oyster catchers, turnstones, and other birds that are foraging in the intertidal, they have more food available when there are no sea stars. But it's kind of risky because, as I said, there's limits to predation. Like mussels can get so big that nothing can eat them anymore. And I've seen mussels that are like a foot long, and there's really nothing that can eat them because they're just so big. So, I mean, in one sense, it might be good in the short term for vertebrate predators like birds, but in the long term, it would be bad to
No one's really
have
looking
the
at
sea
it
stars
right
looking at.
now. No one's like really looking at it right now specifically.
Yeah, we have another experiment where we are assessing the impact that large predators like birds and crabs can have in the intertidal in the absence of sea stars. And those data are still being analyzed too. So we have a very large lab with many experiments ongoing. There's lots of active projects, and that's another one of them. Yep. Any more questions? Yes.
It's so great that you get to be a part of these experiments that have been going on for so long. And at this point, now that you've been involved in it for a while, is there like any questions that you've kind of formulated yourself separately that you personally would like to have answered?
Yeah, I designed a study, another experiment, because my lab is all about field experiments, which is really the best way to know a system well, is to test it, test your hypotheses. And in my study, I'm investigating the interactions between mussels and surf grasses, which are two of the most important foundation species in the intertidal. And I'm looking at how they survive with and without sea stars to eat the mussels. So I've transplanted mussels, which is relatively easy to do. You just like pop them off the rock, you put them somewhere else, cover them with a mesh, and then they'll reestablish on the rock. So I've transplanted them in different habitat types, inside and outside of surf grass. And then I examine how well they survive over time. And generally the mussels have survived better in the surf grass at areas where there's higher sea star abundance. But given enough time, the sea stars will eat all of the mussels in surf grass, which is a little bit lower than they normally grow. And I was investigating that because, as you saw in some of those photos, there were some changes from surf grass to mussel-dominated communities. And I was wondering if I could trigger that same kind of community transition, but I can't actually. And that's a good thing, because the sea stars are back. They may not be as abundant as they were in the 90s, but at least they're coming back in terms of biomass and abundance in their relative to like 2,000 populations. So that's one of the questions that I've had. I have a few more dissertation projects, which I'd be happy to chat with you more about after. Yeah. Anybody else? Okay, yeah, one more?
In the beginning, you said that some of these sites were near marine reserves, adjacent to, I'm sure there's multiple studies going on. Do you see any same effects that will happen to the intertidal zone, those affect those marine reserves, or do you see that trend outside of, say, climate change, or are they such different dynamic systems that you won't see that?
Yeah, it's a good question. I think most of the marine reserve assessments have been subtidal, not intertidal. And so we're talking about a very different study system there, a very different ecosystem where you've got fish and kelps and different sea stars and other invertebrates. And I think the marine heat wave likely damaged a lot of, or killed a lot of kelps. But bull kelp is an annual organism, so it's able to be established fairly well following disturbance. That's how its life history is. Whereas giant kelp is a perennial. So bull kelp gets ripped out annually by these large storms, and it's evolved to establish itself after disturbance pretty quickly. So I haven't looked at the literature or the data on the subtidal assessments that are done in marine reserves, so I can't really say what the impacts of sea star wasting disease or marine heat waves are. But that would be my educated guess, that the marine heat waves did damage kelp, at least initially, but there's good potential for them to reestablish because they're annual plants, they're annual algae. Okay, well, thank you all for coming.